Hi everyone, my name is Susan Eshelman and I'm a PhD researcher at the University of Edinburgh. And today I'm going to talk about my research focusing on the Malagasy grassy flora and understanding the link between environment and functional traits. Grasses are a dominant feature of many terrestrial ecosystems, and they can tell us a lot about the processes going on within these ecosystems. So here you can see 5 examples of ecosystems, all with different grassy layers. And these grasses are subjected to different environments and therefore have a variety of structures. So within our study, we aim to improve the basic understanding of the factors that influence grass structure, distribution, as well as ecological strategies. Currently, within functional trait ecology, things are either done on the study site plot level with individual trait measurements, or on larger scales using databases such as TRI or GrassBase to do large-scale analysis. And we noticed a bit of a gap in terms of study scale, as well as a lack of studies focusing primarily on grassy functional traits. Therefore, our study aims to address this gap by utilizing Malagasy herbarium specimens to look at grass traits on an island-wide scale. To do this, we measured over 1,600 individual grasses from the herbarium collections at Kew Gardens and the Paris Natural History Museum. And here you can see a distribution of our sampled individuals. We decided to work with Malagasy grasses for a few reasons. Firstly, Madagascar is an island, so it's a contained environment and can act as a single study unit. Madagascar also has a highly diverse grassy flora with a large number of endemics, which is rare for an island of this size. Madagascar has also undergone relatively recent transformations with the loss of megafauna, and this has led to a lot of questions about community assembly across the island. And lastly, the Kew Gardens and Paris herbariums both work closely with the Kew Madagascar Conservation Center. Therefore, we know a lot about the taxonomy and phylogeny of the grassy flora there, as well as have a large collection of specimens from across the island. And because of this, we are actually able to sample a lot of the different types of grassy environments I showed earlier. Therefore, with our study, we explored 2 main questions. First, which environmental characteristics such as temperature, water availability, disturbance influence plant functional traits related to growth and resource acquisition, and in what direction? And then secondly, once we understand these relationships, can we predict how traits vary across the island, given patterns and underlying drivers? To do this, we first hypothesized based off the literature and our own knowledge what the key environmental drivers are that influence grass properties. And from there, we identified proxies for these environmental drivers as well as properties of interest. We then measured our set of functional traits off of our herbarium specimens and then extracted environmental variables from the locations where those specimens were collected. Then, to test our hypothesis of trait responses to environment, we fitted a family of model for each trait with our environmental variables as predictors. And then from these models, we predicted mean trait values across the island. So as I just mentioned, we first explored the literature to identify key environmental drivers that influence plant properties related to ecological strategies, which are listed here. So, for example, it's well documented that grasses in areas with low water availability possess tolerant strategies such as low leaf area, which help them conserve water. And so in order to measure and test these relationships, we picked environmental variables that can act as proxies for these main drivers and functional traits that reflect the plant properties we're interested in, as well as are sensitive to these environmental proxies. For our functional traits, we measured 5 structural traits and 1 chemical trait off of the herbarium specimens. And these included inflorescence height, which was taken as the distance from the base of the culm to the top of the inflorescence. culm diameter, which is the diameter of the stem, leaf length, which was the measurement from the base to the tip of the leaf, leaf width, which was the width at the widest portion of the leaf, leaf thickness, which was the thickness at the same point that we measured with. Lastly, we removed 1 to 3 leaves off of our specimens to measure leaf nitrogen. Leaf nitrogen was only taken for a subset of specimens due to the availability of leafing material on the herbarium specimens. When we look at these traits in a principal component analysis, we can see that they are able to capture a lot of the variation in grassy structures across the island. So along this x-axis, you can see that height, culm diameter, and leaf length drive a lot of the variation here. And to help you visualize these structures, here you can see that grasses are taller with more narrow, long, thick leaves and low nitrogen, such as this trachypogon. And then on the other side, we have our low-lying grasses with shorter, wider leaves, such as this brachiaria. And when we compare our principal component analysis to previous studies looking at plant functional trait relationships, such as Sandra Diaz's 2004 paper, we can see that the trait relationships we found in our herbarium specimens are very similar to the same relationships seen in live specimens. So, for example, plant height drives a majority of the variation on the x-axis for both PCAs, while traits related to resource acquisition drive the variation on the y-axis. And these similarities and relationships emphasize the usefulness of herbarium specimens to study traits. So once we collected our traits, we then extracted 5 environmental variables from collection sites for each individual. And these variables act as proxies for the environmental drivers I discussed earlier. And here you can see 2 PCAs of the Malagasy environmental space, with the gray points representing the entire island's climatic space and the blue points representing our data. And as you can see, our environmental variables capture 75% of the variation within the first 3 axes. Now that we have traits that capture the variation of grass structures and variables that represent our range of environments, we can move forward with our hypothesis testing. We specified our hypotheses to how we expect each trait to vary with each individual environmental predictor. And for the purposes of time, I'm just going to focus on inflorescence height, which we know is one of the main drivers of that first axis of variation in grassy traits. So for inflorescence height, we expect a positive relationship with mean annual temperature and yearly mean burned area, and a negative relationship with aridity and our global human modification index. We didn't have any preconceptions about inflorescence height's relationship with percent sand. For our modeling, we used a phylogenetic linear mixed model and utilized this FIRE package, which allowed us to control for phylogenetic non-independence and spatial autocorrelation. To model the trait environmental relationships, we fit a family of models with a single trait response and with combinations of our environmental predictors. Weighted AIC was then calculated and used to rank our models. Results I show here are the consensus relationships observed in the top ranked models up to a cumulative AIC weight of 0.9. And these models are all strongly supported with an R-square close to 0.78. So for inflorescence height, we found that our hypotheses were partially true we could confirm our first two hypotheses, that mean annual temperature and yearly mean burned area did have a significant positive relationship with inflorescence height. We also saw something we didn't predict, which is that percent sand had a significant negative relationship with inflorescence height. What we did not find, however, were significant relationships for our global human modification index and our aridity index. Overall, these results shown are partially in line with our previous understanding of temperature and fire influence on grass structure. They also highlight that soil drainage and nutrient availability might have a bigger influence on grass structure than previously thought. After this, we used model-weighted predictions to estimate inflorescence height across Madagascar. So what you see here is a map where each pixel is 1 square kilometer and shows the mean grass inflorescence height regardless of species. We limited our prediction within the range of the environment sampled, so the white areas on the map represent areas outside of these ranges. And we can now compare this map to the vegetation biomes map. And you can see that our single trait, inflorescence height in this case, shows a more complex pattern across Madagascar than the biome classification does. This, along with predicted distributions of our other traits, are likely to provide additional dimensions of complexity for these spatial patterns. Analysis of spatial patterns across the trait dimensions could help to generate a more resolved map of grassland habitats in Madagascar. Lastly, these models allow us to predict future distributions of grass traits in Madagascar in response to environmental change. So in summary, our study shows the utility of herbariums to explore trait environmental relationships in grasslands. Moving forward, this work can be used as a framework for evidence-based modeling of traits and predict trait responses to climate change. I want to extend a special thank you to my collaborators who helped me put all this work together, and I look forward to any questions.